Hi class, I'm Natalie.
I'm Ben.
And I'm Chris, and we're here to teach you about gene editing.
Today's topic is talons.
Let's start with some background information.
Hey class, do you know what a monkey, an apple, and a dolphin all have in common?
They all contain DNA.
DNA is the blueprint that the cells in our body follow to build the different tissues that make up our body. Different DNA makes a different blueprint, different things are built. That's why the cells in our body are different from cells in an apple. They have different DNA.
The blueprint the DNA codes for is determined by the pairings of 4 nucleotide bases. These bases are named thymine, which pairs with adenine, and guanine that pairs with cytosine. This pairing is called being complementary.
These base pairs are read by cells in groups of 3 called codons. Several of these codons in a row make up a gene, which is later transcribed and translated into proteins which affect the cells in different ways.
In this video, we're going to show you how the order of these nucleotides determines everything a cell does, and how changing the order through gene editing can be used to benefit people.
Gene editing works a lot like video editing. Say we want to delete a dirty word from a movie, like underpants.
Hey, your food smells like underpants.
What? I really like this food.
First, we identify the beginning and the end of the part of the movie that we want to remove, and then we make a quick cut at each end, and it's like the deleted portion was never there. We can make these edits to DNA in the same way.
All gene editing techniques share the same components and steps. There is a binding domain which matches a nucleotide sequence on the DNA and helps the gene editing complex physically associate with the DNA. And there is a nuclease domain. The nuclease domain is what actually cuts the DNA and allows us to insert and remove nucleotides.
Modern technologies are making it easier and easier for us to actually edit this biological code, creating things like glow-in-the-dark cats and cows that fart less, and learning even more about how life works.
A recently discovered tool known as talons helps us to do just this, removing and editing genes just like a director editing scenes in a movie.
The first part of the talon complex is called the binding domain, which locates the part of a sequence we want to target. If the gene we want to cut starts with the sequence CAT, we design a talon that is complementary to these bases. The other half of the talon makes the cut. Something that cuts DNA is called a nuclease, and the name of the nuclease we use is FOC1. Each talon is engineered to recognize different sequences, but FOC1 is exactly the same for every talon. It just cuts. Since DNA is made of 2 attached strands, each FOC1 only cuts 1 strand. We actually use 2 talons to make each cut. One talon recognizes the location on one strand, and another recognizes the opposite. When they meet together, they activate and make the cut.
To cut out a gene, the 2 ends are essentially glued together. Hey, your food smells like...
What? I really like this food.
Or we could replace that gene with a different one. Hey, your food smells good.
What? I really like this food.
It's also possible to target and change single bases, a technique that could someday be used to repair diseases that are caused by small genetic mutations.
Now let's take a look at how gene editing technologies have been put to use.
There are a lot of cool ways that talons can or could be used in our everyday lives, from engineering crops that are drought resistant to helping people with genetic disorders.
Some of the ways talons can be used are in model organisms, cell-based disease agriculture, and therapeutics. We'll start by talking about model organisms.
Model organisms are important because they allow us to study something like a mouse and apply that research to humans.
One study used talons to induce a Rett syndrome-like condition in monkeys by editing their genome during development. This is important because researchers could then study the neurological condition on non-human patients.
Using model organisms such as in this example, we can make more progress towards understanding the disease and helping humans that are affected by it.
We can also study human diseases using cell-based disease modeling. This is when we use cells derived from humans with genetic disorders and manipulate them to see how different edits affect the cells. Talons can also be used in agriculture to make crops herbicide resistant and pigs resistant to pathogens, for example.
Herbicide resistance is important because it means that farmers can spray their fields to kill weeds, but keep the things that we eat and use safe. We can help farmers from having sick pigs by engineering the pigs to be resistant to a pathogen for which there is no cure or vaccine.
Talons can also be used in gene therapies, letting us cure people with genetic diseases by actually changing how genes are expressed in their bodies.
For instance, some people have blood cells that look like this. when really they should look like this. Having blood that looks this way can cause some serious problems, but using talons, we can possibly edit the gene causing this problem and fix it.
So we've learned a lot about DNA today, and so we're going to do a quick review to make sure you've got everything down.
Most gene editing techniques are very similar in the way they work. A binding domain finds complementary base pairs along the DNA, binds with it, and a nuclease domain makes a cut. This can be used to remove or insert different genes, changing the blueprint that cells read off of.
Gene editing is used in a lot of different ways, from creating crop varieties that we eat every day to helping researchers study diseases. It can even be used to make glow-in-the-dark animals.
Even though we've been focusing on talons as a gene editing technique, it is important to realize that it's not the only approach we have to editing genomes.
Like we mentioned before, most of the procedures for editing are the same, but some are more efficient than others. One of the first techniques used was zinc finger nucleases, which was replaced by talons, which has effectively been replaced by CRISPR-Cas9 system.
The reason for replacing previous techniques is that some are more efficient in certain systems than others.
Currently, a lot of these technologies are limited by the inability of scientists to effectively deliver the gene editing molecules to the correct cells and confer the changes we want. Thanks for watching!